Hello and welcome to my channel. My name is Shirsha and in this particular lecture series we are discussing about protein purification. So as I have already stated in the previous lecture, since protein purification is a very vast topic and it would not be possible to complete all of it in one go, otherwise it would become too hectic and difficult to grasp. So we are going to cover this entire concept in a series of lectures. I've already uploaded the first lecture in this particular series, which was about the different techniques of cell lysis, what are the advantages and disadvantages of each of these, when to select which, and the basic principle of each of these methods. So in this particular lecture series, we are going to talk about the next step, that is what to do now that you have lysed your cells. So why was cell lysis important? A brief recapitulation. Because your protein of interest will be expressed by the cell and will be residing within the cell. So in order to release this protein from within the cell, it is imperative to lyse the cell. Now when lysing the cell, it is crucial to keep certain points in mind. If I briefly recapitulate, the primary point of concern is to ensure that A, your protein is indeed released from the intracellular compartment where it is stored post synthesis by the cell, and B, during the lysis process, your protein does not undergo denaturation or your protein is not damaged in any way. And how, you also have to keep in mind that how your protein's subsequent purification steps should not be affected by the choice of lysis method. So with that in mind, now that we have lysed the cells, we are going to move on to the next step, which is isolating the load. So once you lyse the cells, you will have your intracellular contents that are released in the solution, and your solution will also contain cellular debris. So it is very important to remove the cellular debris so that now you have what is left behind, what is known as the lysate. That is what you obtain post lysis of the cell. So once you have the lysate, you will and from which you have already removed the cellular debris. So now what you have is referred to as the load. What is the load? Now your protein of choice which you will be purifying will probably have some tag with the help of which you will purify it, or you might make use of its physicochemical characteristics, like for instance its overall charge to purify. So for that purpose, you will be requiring a column which has special beads which can either bind to the specific tag which your protein of interest has, or which can bind to your protein based on its specific charge. So on these beads, you're going to load your lysate, and therefore this clarified lysate. What do I mean by clarified lysate? Lysate from which the cellular debris has been removed, lysate from which the genomic DNA has been removed. This clarified lysate will now be known as your load, because this you're going to load onto these beads. Okay, so in this particular lecture we are going to talk about how to isolate the load. What should be your next step once you have lysed your cells? So one common method that is typically followed is precipitation and differential solubilization. Now, what do we mean by this? In bulk protein purification, a common first step to isolate proteins is precipitation with ammonium sulfate. Now, this ammonium sulfate precipitation is a very common technique, and I'm pretty sure you must have heard of it somewhere or the other when studying about protein purification. So this is performed by adding increasing amounts of ammonium sulfate and collecting the different fractions of precipitated protein. Now what do I mean by different fractions? Suppose you add a certain percentage of ammonium sulfate. Let's say for argument's sake 20%, and you get a certain level of precipitation. So you collect it. Now you increase the amount of ammonium sulfate you're adding, say to 30%, and now a different population of proteins get precipitated. You collect this. So as you gradually keep on increasing the amount of ammonium sulfate or the percentage of ammonium sulfate you're adding, more and more proteins will get precipitated, and these different fractions are then collected. Now, how does this work? What is the mechanism behind this? Or how does the addition of ammonium sulfate promote precipitation. We will discuss that. So subsequently, the ammonium sulfate can be removed using dialysis. So say for instance, you have precipitated out as many proteins as you possibly could without precipitating out your protein of interest. So your protein of interest is still in solution and unwanted proteins have been precipitated out. So now you have to remove this ammonium sulfate from the load before you can, add this load to the beads which you'll be using for purification. So this ammonium sulfate can be removed by simply performing a dialysis. Now during the ammonium sulfate precipitation step, hydrophobic groups, that is the water hating or the water repelling groups present on the proteins are exposed to the atmosphere, attracting other hydrophobic groups. So because these hydrophobic groups get exposed, hydrophobic groups have a tendency of interacting with other hydrophobic groups. Why? Because they don't want to be exposed to solution. By their nature, it is their inherent nature and it is suggested in their name. They hate water. They are scared of water. They are hydrophobic. So they will try to sequester themselves away from water. And in order to achieve that, what these hydrophobic groups do is that they prefer to interact with other hydrophobic groups. And this results in the aggregation of the protein, which subsequently will lead to precipitation. So this results in formation of an aggregate of the hydrophobic components, and the protein precipitate is typically visible to the naked eye. You can visualize it in the form of white flakes floating around in your solution, and if you give it a quick spin, if you centrifuge it at high speed, these flakes of protein precipitates typically settle down and you can clearly and easily visualize them with just your naked eye. Now, one advantage of this method and one major advantage that too is that it is pretty cheap. It can be performed inexpensively, even with very large volumes, because all you need to perform this method is ammonium sulfate and knowledge of how soluble your protein is, and at addition of what percentage of ammonium sulfate will not affect the solubility of your protein of interest. That is, your protein of interest will remain in solution. If you have these two basic ideas with this one basic idea and access to ammonium sulfate, you can perform this particular method, and it is very easy to do so with even larger volumes. So therefore, if you're thinking about production of any particular protein for pharmaceutical purposes, this can be a very suitable first step. So this precipitation of proteins by adding ammonium sulfate out of solution is called salting out. Why salting? Because you're adding the salt ammonium sulfate, and out because your protein of not interest, but these other non-interest proteins or contaminating proteins that you want to remove are brought out of solution. They were in solution when they were soluble, and when they precipitate out, they are brought out of solution, and hence we call this salting out. So say for instance you have your target proteins, right, and you precipitate out first these red ones and then these green ones, and finally you have just the yellow ones, which is your protein of interest in the solution. So let's take a deeper look at how ammonium sulfate precipitation or salting out actually works. Let's look at its mechanism. So at low ion concentrations, that is less than 5 moles per liter, the solubility of proteins increases with increasing salt concentration, an effect termed as salting in. So salting in is the exact opposite of salting out, whereby you are increasing the solubility of your protein in the solution. Now, as the salt concentration is further increased, the solubility now begins to decrease. So, at low ion concentration, you increase the salt concentration, so your solubility of protein increases. But after you cross a certain point, now when you increase the salt concentration further, the solubility of your proteins decreases. At a sufficiently high ionic strength, the protein will precipitate out of the solution, and this is what we term as salting out. So now your protein of interest gets salted out of the solution. When the ammonium, that is a NH4 plus, I'm sorry, there has been a typographical error. The 4 should be a subscript on H. Let me quickly try to correct that. Okay, yeah. So we move the plus from here and we have the plus 4 here. Okay, cool. So when the ammonium, that is NH4 plus, and sulfate SO4 2 minus ions are within the aqueous solution, they are attracted to the opposite charges that are evident on the compound that is being purified. So this is like pretty straightforward and easy to understand, and it makes logical sense. You have positively charged ions that will get attracted towards the negatively charged regions of the protein that are exposed on the surface, and when you have these negatively charged ions, they will get attracted to the positively charged regions of the protein that are exposed. Now, this attraction of opposite charges prevents the water molecules from interacting with the compound being purified. So, let us backtrack for a moment. What ensures that your protein of interest is soluble in water? What do we exactly mean by its solubility? By its solubility, or by stating that a protein is soluble in water, what we mean is that the protein molecule has a water shell or a hydration shell surrounding it. That is, there are water molecules all around the protein interacting with the charged residues exposed on the surface of the protein, ensuring that the protein is remaining within solution. Now, when you're adding increasing amounts of these two ions, ammonium and sulfate, these ions will go and interact with the charged residues on the surface of the protein. And because these oppositely charged residues, when interacting, their strength of interaction will exceed the strength of interaction between the water molecules and protein. So as a result, they will displace out the water molecules and they will take up their place. And this will remove the hydration shell from the surface of the protein that was otherwise stabilizing the protein in solution, ensuring the solubility of the protein. And this would lead to its salting out. So this image will help better understand it. So you have this protein molecule. It has these blue patches, which are the hydrophobic regions on its surface. Now, when you're salting in at this particular level, that is at the low ion concentrations, when you keep increasing the salt concentration up to a certain level, that is up till this point, it will increase the solubility of your protein. So you have these hydration shell surrounding the proteins, which is essentially the water molecules interacting with the protein, and you have these hydrophobic patches that interact with each other and therefore bury themselves within, and they are not exposed to water. And you have some of these iron or salt molecules on the surface, which overall stabilizes the solubility of the protein. Now this, as you're increasing salt concentration, solubility is increasing up till a certain point, till here. Then you, till here, and then you reach this equilibrium point, or rather the tip of the bell-shaped curve, where you have an optimum amount of ions in the solution because of the salt that you have added, and they are stabilizing the protein. And in fact, like if you compare this image with this, over here you can see that different molecules are interacting with each other through these hydrophobic regions. And when you are increasing the salt concentration, then the different protein molecules can separate out, because these exposed hydrophobic regions are not really exposed, but rather these blue patches, they are covered up by the salt molecules, and therefore this is increasing the solubility of your protein. Why? Because instead of clumping up like this, or instead of like three molecules in this case having to exist together, you have individual molecules existing independently. So here the solubility is at its peak. But now when you keep increasing the salt concentration even further, so what these salt molecules do is that they cover up the entire surface of the protein, removing all the water molecules. And now because all the water molecules have gotten removed, the protein is no longer in the solution, but rather it has come out. And this is how your protein gets salted out. I hope I have been able to clarify that. So purification of integral membrane proteins. So we are done dealing with sorting out. Next, we're going to move on to differential solubilization. So purification of integral membrane proteins. So what do I mean by integral membrane proteins? Proteins that are embedded within the cell membrane, that are a part of the cell membrane. So in order to purify such proteins, you first have to disrupt the cell membrane in order to isolate any one particular protein from others that are in the same membrane compartment. So a membrane is a continuous layer, and say you have your protein inserted in the membrane. So you should be able to break apart your membrane without damaging your protein which is embedded in the membrane. So therefore, when you're breaking apart the membrane, your method of choice should be a very mild method. You can't, like, in the previous lecture, if you have gone through it, we have discussed about how we can use detergents to lyse the cells, to solubilize the membranes. But your detergent of choice, in case you want to isolate a membrane protein, has to be a mild one. You cannot select any harsh membrane which will completely denature your protein which is embedded in the membrane. So you need to select an ideal detergent for this purpose. So sometimes a particular membrane fraction can be isolated first, such as isolating mitochondria from cells before purifying a protein located in the mitochondrial membrane. So yes, if you have a protein which is expressed in the membrane of the mitochondria, it would always be preferable if you can isolate the mitochondria first and then you move on to isolate the membrane protein. So what would be the advantage of such a scenario? Like, because if you imagine isolating the mitochondrial membrane protein from the whole cell without purifying the mitochondria first, then you would have the cell membrane as well, which will be interfering with your subsequent purification steps. So first, isolating the mitochondria from the entire cell would help in further steps of purification. It will make things easier. And how can we isolate just one organelle out of a cell? We will discuss about that in the upcoming slides. So a detergent such as SDS can be used to dissolve cell membranes and keep membrane proteins in solution during purification. But because SDS is very strong as a detergent, and therefore it will cause denaturation of your membrane protein, SDS is not used, and milder detergents like Triton X100 or Sharps can be used to retain the protein's native conformation during complete purification. Because if you're purifying a protein, you would either want to do some functional assays with it, or you would want to proceed for structural studies, or you simply want to purify the protein to use it as a biopharmaceutical compound. So it is imperative that you are able to maintain the native conformation, that you are able to preserve its fold, because ultimately a protein's function is determined by structure. So in order to do that, avoid detergents like SDS and milder detergents like Triton X100, Sharps, or even more sophisticated detergents like LMNG or DDM or GDN. These can be used. So what happens is that you have your membrane over here and you have this transmembrane protein, or you can have this integral membrane protein. Like both of these are integral membrane proteins. This is a peripheral membrane protein. So when you add the detergent at low CMC, that is critical micellar concentration, these detergent molecules insert themselves in the membrane at different spaces. And when you further increase the concentration of the detergent, you end up with a mixture. You end up with a mixture of what? Of this transmembrane protein encapsulated in a detergent micelle. You have this peripheral membrane protein again encapsulated in a detergent micelle, and you have just free detergent micelles and also detergent micelles with the lipids, that is the phospholipid from the cell membrane embedded in them. So what is CMC? CMC is the critical micelle concentration. It is defined as the concentration of detergents above which micelles are spontaneously formed. So this is your micelle. CMC is the concentration beyond which such micelle formation occurs spontaneously. So if you look at this curve of detergent concentration versus, detergent concentration versus detergent monomers in solution. So when you increase the concentration of detergent up until the CMC, you have monomers in the solution. And once you reach CMC, you get spontaneous micelle formation. And now the detergent monomer concentration becomes constant. And now you will get micelles. So the next step that we are going to discuss about is centrifugation. That is, once you have prepared the lysate, that is, you have lysed your cells and you have done all sorts of precipitation. So centrifugation, let me backtrack. Centrifugation you can perform directly after cellular lysis to remove the cell debris and to clear the lysate, and you can perform it as well after your ammonium sulfate precipitation, provided you're doing ammonium sulfate precipitation. But in case you are doing ammonium sulfate precipitation, before you do it, you have to perform centrifugation to remove the cellular debris, and only then will you do ammonium sulfate precipitation and then another round of centrifugation to clear your load, the final load. So centrifugation is a process that uses centrifugal force to separate mixtures of particles of varying masses or densities suspended in a liquid. So a little bit of 10th standard physics. What is centrifugal force? It is the force that is acting on a particle when it is rotating about a fixed point or about an axis. So centrifugation utilizes this centrifugal force to separate out mixtures of particles that differ in their either mass or density. When a vessel, typically a tube or bottle containing a mixture of proteins or other particulate matter such as bacterial cells, is rotated at high speeds, and by high speeds I mean very high speeds, the inertia of each particle yields a force in the direction of the particle's velocity that is proportional to its mass. So heavier the particle, greater would be the force exerted on it, and quicker it will settle down, or it will settle down at the bottom of this vessel, the tube or the bottle. So the tendency of a given particle to move through the liquid because of this force acting on it is offset by the resistance that the liquid exerts on the particle. So if your particles are trying to migrate towards the bottom of the vessel because of this force, then the resistance will act in an exactly opposite direction because of the liquid that is present, and this will offset this force. The net effect, that is, if you subtract the force acting towards the bottom, if you subtract from that this resistance, that is the force acting towards the bottom minus the resistance, that is the net effect of spinning the sample in a centrifuge, is that massive, small and dense particles move outward faster than less massive particles or particles with more drag in the liquid. So if you have something which is more compact, that will face less drag and that will settle down quicker. And if something is heavier, then also it will settle down quicker. Compared to that, if you have something lighter or something that is more diffuse or spread out, that will settle down slower. So when suspensions of particles are spun in a centrifuge, a pellet may form at the bottom of the vessel that is enriched for the most massive particles with the least drag in the liquid. So this is what a typical centrifuge looks like, and these pink things that you can see are basically the lids of several tubes, and each of these adapters are holding like almost 40 to 50 tubes depending on the capacity of the particular centrifuge that you are using. So this is what happens. You have in solution your particles, that is the black dots, suspended. So your solute particles are evenly distributed in the solution. And let us assume this is the center of rotation and this is the distance, that is the further you go down towards the bottom of the vessel, greater is the distance from the center of rotation. So the net forces acting are, you have the centrifugal force that is acting in the downward direction, which is proportional to the mass of your particle, and then you have the bind force of the solution and the frictional force which oppose the centrifugal force. So if you subtract the bind force and the frictional force from the centrifugal force, you will get the net force acting on the particle, which will be responsible for pelleting it down. So with the passage of time, your solute particles get distributed towards the base, more and more towards it. So the force acting on a particle, so now we will do some maths, some very simple and basic maths. The force acting on a particle during centrifugation, that is F, is given by F equals to m omega square r, where m is the mass of the particle, omega is the angular velocity in radians per second, and r is the distance of the particle from the axis of rotation. So this is a very simple plus 12 plus 2 level physics formula that we are all aware of, related to angular motion. Now, usually the value given for the force applied to particles during centrifugation is a relative one. So it is very important to keep this in mind, that the force applied during centrifugation, the value of it is not at all absolute. It is relative. It is compared with the force that the Earth's gravity would have on the same particles. So this is called the relative centrifugal force, or RCF. So if you are reading any protocols online, then you would come across this term, the RCF, or even if you go through manuals of these different centrifuges, you would come across this term RCF. So that is the relative centrifugal force, which is the ratio. Okay, fine. Let's go through the formula. It is given by centrifugation force divided by force of gravity. So it is a ratio of the force that is acting on the particle as a result of the centrifugation process to the force that the Earth's gravity would be exerting on that particle. So if you put in the formulas, you have m omega square r divided by mg, that is mass into acceleration due to gravity. You cancel out mass, you get omega square r by g. So g is the acceleration due to gravity. So omega square by g, you can further expand it, where the omega square, or the angular velocity, would be given by 2 pi time multiplied by the RPM, that is the rotations per minute, divided by 60, and you're squaring on that, divided by 980, which is simply the value for acceleration due to gravity. And finally, when you solve this out, you get this particular value. So if you want to find out the relative centrifugal force acting on any particle, it is now independent of the mass of the particle. Why? Because the mass of the particle has been cancelled out by the force of gravity exerted on it, and it simply depends on the RPM. So now the mass of particle has no role to play on determining the relative centrifugal force that is acting on your particles. It is simply dependent solely on the rotations per minute, or the RPM. So RCF is customarily followed with the symbol G, indicating that the applied RCF is a multiple of the Earth's gravitational force, because you are dividing it by the Earth's gravitational force. So simply whatever RCF you're getting, that is a multiple of the Earth's gravitational force. So if you now think about it, if you have read any protocols, you would see it is mentioned that spin your samples at so and so G speed, that is spin your samples at 11,000 G RCF. So now you know what RCF means and what this G means and why it is there. And even in centrifuges, if you're working in a laboratory and if you have access to a centrifuge, you can see that there are two modes of setting the speed. You can either set it in RCF or you can set it in RPM, that is the rotations per minute. So this is what it actually means. Now, RCF is independent of the properties of the particles being sedimented. So, therefore, you can set the speed in RCF, because if it was dependent on the properties of the particle, you couldn't have a uniform RCF that you're setting on the centrifuge, the machine. However, a particle's sedimentation rate, that is the rate at which it will settle to the bottom of the vessel at a particular RCF. So RCF is the relative force you're applying, but how fast or how slow that particle will sediment depends on the properties of the particle. So RCF is independent of the properties of the particle, but upon applying a specific RCF to your particles, how quickly or slowly your particles will settle down depends on the properties of the particle itself. The instantaneous sedimentation rate of a particle, that is at any point of time, the sedimentation rate of a particle during centrifugation is determined by number 1, the centrifugal force, number 2, the bind force of the medium, that is the force exerted on it by the solution in which the particles are suspended. Number 3, the frictional resistance to the particles movement. So these are all points that we have already covered in 2 slides back. So the sedimentation coefficient, or S, is the rate at which a particle sediments under conditions of unit acceleration, that is S is dr dt omega square r. So the Sedberg unit, which is named in honor of the scientist who came up with this entire concept, abbreviated as capital S, is used to describe sedimentation coefficients and is equal to the constant 10 to 13 seconds. Large values of S, that is faster sedimentation rates, correspond to larger molecular weight. Dense particles therefore sediment more rapidly. So if you think of the ribosomal subunits, they are the eukaryotic ribosomal subunit. So you'd have the 40th subunit and the 70th subunit. So this S actually comes from the Swedberg unit, which is a measure of its density and how fast it will settle down when centrifuged. So now, taking a look into the technical aspects, there are 2 types of rotors that are commonly used in centrifugation. We have the swinging bucket rotor. So as the name suggests, in swinging bucket rotor, you have a series of metal buckets for containing the sample tubes, which are attached to a central harness. And during the acceleration process, the buckets swing upwards from a vertical to a horizontal position, and during deceleration, the buckets swing back to the vertical position. So basically you have in a setup similar to this in case of a swinging bucket rotor. At rest, your buckets are straight, that is they are vertical. When you start spinning, because your buckets are attached via a harness, so when you are spinning them, these buckets will swing up and they will take up this horizontal position. So this is what you get in case of a swinging bucket rotor, and they are very common. Next, you have your fixed angle rotor. So in fixed angle rotor, the buckets do not swing from a vertical to horizontal position, but rather the tubes are maintained at a fixed angle, usually of 15 to 45 degrees from the vertical throughout the centrifugation process. And the radially sedimenting particles, they will basically strike the tube wall and are carried to the bottom of the tube. So this is how things look in a fixed angle rotor. So what exactly do I mean by the radially sedimenting particles? So when we were talking about the forces that are acting on the particles suspended in the solution during centrifugation, we had the centrifugal force acting downwards, and we had the bind force and the frictional force acting upwards. If you think about the diagram that we looked at a couple of slides back, you can go back to just revisit it. Now, essentially what happens is, imagine that this vessel that we were visualizing when discussing about the forces, that the vessel is horizontal. So that is in case of a swinging bucket rotor. when your vessel is horizontal, you have the centrifugal force acting in this direction, that is towards right in this case, and bind force and frictional force acting in the opposite direction. So basically, you have your centrifugal force acting in this direction, and you have bind force and frictional forces acting in this direction. So this is what you achieve in case of a swinging bucket rotor, and this is exactly the same thing that we discussed a couple of slides back. Now in case of a fixed angle rotor, because it is rotating like this, your centrifugal force will still be acting in this direction. Centrifugal force will not change its direction to act in an angular fashion like this. It will not suddenly start acting in this direction. It is still acting in the direction as shown by the red one. It is this direction in which it is acting. So your particles are now radially diffusing out. So when these particles are subjected to the centrifugal force in a fixed angle rotor, they will move out, they will hit the wall of the tube, and then once they hit the wall, they will settle down along the length of the wall to the bottom. So therefore, if you think about the pellet that you obtain in a swinging bucket rotor, the pellet that you will obtain will always be a straight line. When you take out your, say for instance, your falcons in which you are centrifuging, you take them out from a swinging bucket rotor, the pellet will have a straight line because it was having a uniform force acting on it and was just directly settling down at the bottom. But if you take out your tubes from a centrifuge which has a fixed angle rotor, in that case the pellet that you obtain will be slanting like this, with more amount of the pellet on the side that was facing the outer direction and lesser amount of pellet on the side facing the inward direction. So this is actually the justification of why you obtain different kinds of pellets in a swinging bucket rotor and a fixed angle rotor. And personally, I believe a swinging bucket rotor is a better choice than fixed angle rotor, but that is just my opinion. So moving on. Differential centrifugation. So now we are going to just very briefly discuss about the different kinds of centrifugation. So first we'll talk about differential centrifugation. Differential centrifugation is used to separate organelles from whole cells for further analysis of specific parts of the cells. So remember, now we were talking about isolating the mitochondria from the whole cell before we proceed with purification of the mitochondrial membrane protein. So in order to do that, we will be performing differential centrifugation. A sample is subjected to repeated or multiple rounds of centrifugation, each time removing the pellet and subjecting the supernatant to an increasing centrifugal force by increasing the speed. This sequence is repeated till all the particles have been sedimented. Each sediment can then be used for further experimentation. So you have your tissue cells, you have homogenized it. Now you spin it first at a lesser speed, say 800 G. So you have the pellet which is enriched in nuclei and cellular debris, because these being the heaviest, they settle down first at the lowest speed. You collect the supernatant and you spin it again at a greater speed, 20,000 G. So now the pellet you obtain is enriched in mitochondria and chloroplast if the cells are from a plant. You once again take the supernatant, transfer it to a fresh tube and spin it again at a higher speed, 100,000 G, for longer time. Now please note that as you're increasing the speed, you're also increasing the time, because the time for which you're spinning does play an important role in determining your pelleting. So now you have the pellet which is enriched in microsomes, which are pieces of the plasma membranes and cells' internal membranes. You once again collect the supernatant and spin it at a still higher speed of 150,000 G for 3 hours, and now you have the pellet enriched in ribosomes. So this process is also referred to as subcellular fractionation, because you're fractionating the different cellular components. So you're spinning, collecting the supernatant, spinning it again, collecting the supernatant, and so on and so forth. So next we have density gradient centrifugation. A greater degree of purification can be achieved by density gradient centrifugation, in which organelles are separated by sedimentation through a gradient of a dense substance such as sucrose. The particles are layered atop a density gradient and then they're allowed to sediment from this position. Density gradients may be of two types. It can be either discontinuous or stepwise, or it can be continuous. So a discontinuous gradient is prepared by successively layering solutions of decreasing density in a centrifuge tube. So at the bottom you have the solution which has the highest density, and as you go up one layer after another, the density of the solution gradually decreases. In contrast, a continuous gradient is prepared by mixing the dense and lighter solutions at a continuous rate and delivering these mixtures to a centrifuge tube in a continuous stream. So you will not have these distinct steps where the density suddenly changes, but rather you would have a gradual change in density. Solutes selected for a density gradient are selected on the basis of their solubility in water and their minimal detrimental effects on the cellular organelles and other biomolecules to be centrifuged. So it is to be kept in mind that whatever solute you are selecting for generating the gradient, that should in no way be detrimental to your protein or to your sample. It should be an inert solution. Sucrose, ficol, which is a polymer of sucrose and epichlorohydrin, and porcol, which is a colloid formed from silica, are commonly used. So you will commonly hear these terms: sucrose density gradient or a porcol or ficol density gradient. So these are the different types of gradients. You can have a density barrier where you essentially have two solutions having different densities. So this is your barrier. So you're moving down, 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 and boom, there's a barrier. You have a more dense solution. You can have a discontinuous gradient where as you're moving further down the tube, you have layers of increasing density. And then you have the continuous gradient where there are no such clear demarcating layers or segregation, but a uniform increase in density as you're moving down from the top to the bottom of the tube. So under density gradient centrifugation, we again have two more types that we are going to discuss. The first one is rate zonal centrifugation. So what do I mean by it? In rate zonal centrifugation, the starting material is layered on top of the sucrose gradient, or it can be any other gradient. Particles of different sizes sediment through the gradient at different rates, and they move in the form of discrete bands. So particles of a specific size will group together to form a discrete band. Particles of a different size will group together to form a separate independent discrete band. And these particles move in the form of these discrete bands through the gradient. If centrifugation is allowed to proceed for long enough, all of the particles will eventually reach the bottom. So in rate zonal centrifugation, the duration of centrifugation plays a very important role. However, if the duration of centrifugation is carefully limited, then the particles will be distributed throughout the density gradient in order of their sedimentation coefficients. Both particle size as well as particle density determine the final position of the particles in the gradient. Size is a more important factor and it has greater role to play compared to the particle density. Following centrifugation, the collection of individual fractions of the gradient provides sufficient resolution to separate organelles of similar size like mitochondria, lysosomes, and peroxisomes. So these organelles of similar size, which were otherwise precipitating together in case of subcellular fractionation, they can be separated out by doing rate zonal centrifugation. So essentially what you do is that you separate the particles on the basis of their size. You layer the sample atop of the gradient that you have generated, and this is how the particles are. The sample is before I'm sorry. This is how the different density layers are, or the layers of different densities, and you have your sample at the top. And after centrifugation, these particles you see in the form of bands have separated out. But like I already mentioned, keep in mind that the duration of centrifugation plays a very important role in this separation, and if you do this for long enough, everything will simply settle down at the bottom. The last thing that we are going to discuss in this particular lecture is equilibrium centrifugation. So equilibrium centrifugation is another type of density gradient centrifugation which can be used to separate subcellular components on the basis of their buoyant density. So in rate zonal centrifugation, we were relying on the size of the molecules, but over here, or rather of the particles, but over here in equilibrium centrifugation, separation or resolution of the particles is dependent on their buoyant density, and it is independent of their size and shape. In this procedure, the sample is centrifuged in a gradient containing a high concentration of either sucrose or cesium chloride. Rather than being separated on the basis of their sedimentation velocity, the sample particles are centrifuged till they reach an equilibrium position in the gradient at which their buoyant density is equal to that of the surrounding solution. That is, they reach their isodense or isopycnic position. So this kind of centrifugation is also known as isopycnic centrifugation. So basically you have before centrifugation the different layers which have different densities. So you have your density gradient and you have the sample that you're layering on top. And after centrifugation, you have these different cellular organelles, that is the lysosomes, the mitochondria and peroxisomes, all separated out. Now all three of these have the same size, but because they differ in their densities, they are separated out by equilibrium centrifugation, whereby at this point the lysosome is at an isodense situation. So the forces acting on it in both directions when it is at this point will cancel each other out. So it will stabilize at that particular layer. So in case of equilibrium centrifugation, no matter how long you centrifuge, all of your particles will typically not settle down to the bottom of the centrifuge, of the vessel in which you're centrifuging. So that brings us to the end of this particular lecture, isolating the load, the second step in protein purification. That is, once you have lysed your cells, how do you prepare the lysate for loading onto the beads that you will be using for subsequent purification. We talked about ammonium sulfate precipitation, differential solubilization, and centrifugation, the underlying principle, and briefly about the different types. In the next lecture, we will be discussing about different types of chromatography and how you can use that to purify your protein. So this will be the final lecture in this series where we are going to talk about different types of chromatographies, their underlying principles, and how that can be used to purify your protein of interest, which chromatographic technique exploits which physicochemical characteristic of the protein, which requires modifications, which does not, what are the advantages and disadvantages of each. So I hope I've been able to clear this topic, and if you have any questions or any doubts or any remarks, please feel free to leave them in the comment section and I will definitely get back to you. If you like this video, do like and share and subscribe for more such content. So see you next time. Bye.